Well, good morning, good afternoon, or good evening, depending on where you are joining us from. So, welcome everyone to today's session of the Global IMMUNOTALKs, the series that brings all aspects of immunology to a global audience. Now, today it's my great pleasure to welcome Michaela Gack to deliver us the Global IMMUNOTALK. And just to introduce Michaela, she did her PhD studies in virology at Harvard with Jay Young, where she did some seminal work identifying the role of TRIM25 in activating the key pattern recognition receptor for RNA viruses, RIG-I. And this was actually part of a collaborative graduate training program between Harvard and the Friedrich Alexander University Erlangen-Nuremberg in Germany, where Michaela is actually originally from. And then after her PhD, Michaela moved to the West Coast. She did a postdoc on viral immune evasion at University of Southern California in Los Angeles. And then in 2011, she started her lab at Harvard Medical School before moving to the University of Chicago in 2015, where she was then promoted to professor in 2018. And then in 2020, so most recently, Michaela actually moved from Chicago to the Cleveland Clinic in Florida, where she is currently the Arthur and Marilyn Levitt Endowed Chair and also the Scientific Director of the Cleveland Clinic's Florida Research and Innovation. So Michaela's research focuses very much on the interplay between viruses and the innate immune system and how this may impact immunity as well as the outcome of viral infection. And she has focused much attention on cytosolic sensors of RNA viruses and how host enzymes can play key roles both in the virus life cycle, but also the host response to the virus. And amongst her many discoveries, she showed, for example, that host-derived RNA species, such as transcripts from certain ribosomal RNA pseudogenes, can activate the RIG-I pathway under certain conditions. She looked at the role of various modifications and carried out by host enzymes on both sensors of RNA viruses, including ISGylation of MDA5, but also looking at how host enzymes can impact the life cycle of viruses and can be essential for viruses to complete their life cycle in host cells. And so her approaches also allow us to think about therapies that target conserved host factors, and those might be things that help us circumvent issues such as rapid viral mutation and resistance. And she has received numerous prizes and awards for her research, most notably the Wilczek Prize in 2017 and the NIH Director's Pioneer Award in 2021. And I think just now in 2024, recently, she was elected a Fellow of the American Academy of Microbiology. So as usual, we will have a Q&A session on Twitter or X after the talk, and the instructions will be put up at the end on how to ask Michaela questions. And so in a minute, I'm going to ask Michaela to deliver her lecture. But as usual in this series, I'm going to start by asking her a personal question to be answered before the talk. And Michaela, the question is, how has your view of success in science evolved over the years?
First of all, thanks so much, Catano, for having me today. It's really nice to be here and for the great introduction. To the question, I think the major goal and therefore also the definition of success for my whole lab, is to drive the field, hopefully making discovery that stimulates, more discovery and to some degree also maybe us or stimulating how we think about a field or a certain topic. In ours is, of course, especially innate immunity. So that continues to be, the major goal. When you have your lab for a certain time, I think what also defines the success is how good your trainees are doing, when you see them having their own lab. And I have now multiple, former postdocs who are now running their own lab in multiple different countries. That is, I think, a new measure of success that maybe, certainly 10 years or 15 years ago, I didn't, realize as much as now. And it's a wonderful, experience seeing your own people running their labs and being successful. Yeah.
Very good. Well, thank you, Michaela, for those words. And so now I'm going to invite you to start sharing your screen and deliver your lecture, which is entitled Novel Mechanisms of Virus Sensing in Innate Immunity.
All right, so I'm sharing now my screen. Maybe Caetano, give me an okay when you see the slides.
Yes, it's okay. If you just put it on. There you go. Perfect.
Okay. Maybe one more. Yeah, so thank you one more time, Caetano, for the invitation to talk today about our work on innate immunity. And, before I show you some of our work, I would like to give some introduction briefly. So one can illustrate the human immune response as this pyramid where you have on the bottom, very sophisticated and tailored responses. And that's typically the, adaptive immune response, where certain immune cells, again, in a very tailored way, fight pathogen infections. But on the top of this pyramid, you have the more rudimentary form of immunity, and that's the innate immune system. And my lab really works here on the top or at the tip of this pyramid, which is really the first step of innate immunity, and that is the detection of the pathogen by innate immune sensors. There are obviously many different pathogens. My lab focuses mainly on the detection of viruses. And we are really interested how different sensors, sense these pathogens. What are the PAMs? So what do they recognize? But also how do these innate sensors work? How do they initiate signaling and how are they regulated? We are also very much interested in how do they elicit antiviral effector mechanisms, so what kinds of effector mechanisms in the infected cell, but some of them obviously have more broader and paracrine effects in the infected organism. I am a molecular virologist by training, so we also look at the other side of the coin, I always say. So, viruses, we all know, dysregulate or suppress innate immunity. So I very often say whatever we assay in our experiments and the readouts we have, we always measure the sum of the host and also, the virus that dysregulates any of these responses. We have worked for several years on the RIG-I-like receptors, and this is also the topic of today's talk. So these RIG-I-like receptors are cytoplasmic RNA sensors. They have two primary members, RIG-I and the related sensor MDA5, and they recognize typically viral RNA of many different RNA viruses. So this is kind of the most well-known studied context for RIG-I-like receptors, all these different RNA virus infections. Some of these viruses, such as influenza viruses, are recognized by one sensor, RIG-I, and, for example, coronaviruses by MDA5. But there's more and more evidence that many viruses are co-sensed by RIG-I and MDA5, where maybe the one sensor plays a more dominant role in one infection or type of virus and the other one in other viral infections. But in essence, these RIG-I-like receptors, they signal through a common adapter protein called MAFs. MAF sits at the outer membrane of mitochondria, mitochondrial-associated membranes, as well as peroxisomes, and then initiates a very classical innate immune response via TBK1, tank kinase 1, and then different transcription factors, including different interferon regulatory factors, NF-kappa B. And this then leads to cytokine induction, most prominently type 1 interferon induction, but also other cytokines, including type 3 interferons in many cases. What is not shown here in this picture, but interferons and these cytokines are secreted from the infected cell, and they act through another signaling pathway, either in an autocrine or paracrine-dependent manner, and then upregulate hundreds of interferon-stimulated genes. And the gene products, then, they have very often an antiviral activity through a variety of mechanisms to control virus infection. We have learned over the past several years, actually from the work from many labs, what is a bona fide RIG-I agonist. So how should an RNA, or how does an RNA look like that is recognized and activates RIG-I? Typically, this is a viral RNA, But I tell you in a few minutes, also host RNAs can be sensed by RIG-I in certain contexts. And these bona fide RIG-I agonists have several key features. First of all, a triphosphate moiety, as well as also possible a diphosphate group at the 5' end. And then right adjacent, typically base pairing. So these double-stranded RNA structures that we know for a long time, if you deliver them into the cytoplasm, you get immune activation. We know by now very well how these features of RIG-I agonist engage with the RIG-I protein. So the double-stranded RNA engages with a helicase domain, here shown in purple, the tri or diphosphate group with a C-terminal domain. And this is known to cause a conformational change in RIG-I, exposing these domains, the CAR domain, which then bind to MAFs, this adapter protein, and elicit interferon induction. What I would like to talk about in the first part of my talk is about sensing of DNA viruses by RIG-I-like receptors. This goes really the question back when we started out with that a few years ago, where there was some literature indicating that RIG-I-like receptors, in particular RIG-I, are also involved in the detection of DNA viruses, such as herpes viruses. So DNA viruses means these viruses have a DNA genome. They are very well known to be recognized by DNA sensors. But again, there was in the literature quite some indication that also RIG-I, this RNA sensor, is involved in sensing these DNA viruses. What we did, and I'm showing here some of our earlier work, we used herpes simplex virus type 1. That's a classical herpes virus, so having a double-stranded DNA genome. And we infected DDX58 positive or a knockout mouse embryonic fibroblast. DDX58 is the genome for RIG-I. And what we saw when we infected these cells with herpes simplex virus and measured interferon beta by qPCR in different ISGs, as shown here, you see a significant reduction in interferon transcript induction as well as for the ISGs. Again, indicating that RIG-I, despite being an RNA sensor, is clearly involved in sensing also this DNA virus, herpes simplex virus. We showed, and I'm not bringing here the data here, but we have published that a while ago. So we wondered, what is then the role, RIG-I versus these classical DNA sensors? Many of you might know them, cGAS and I516, are well-demonstrated DNA sensors recognizing herpes simplex type 1 and other DNA viruses. And what we found is that, at least in fibroblast cells, what we looked at, we also looked into epithelial cells, we found that cGAS and I516 acted immediately upon infection within 2, 4, and 6 hours post-infection, whereas RIG-I acts later during infection. We see it being activated around 10 to 12 hours or post-infection. It depends a little bit on the cell type. But in essence, our data suggested that there is a temporal role between RIG-I, this RNA sensor, and these DNA sensors, where the DNA sensors act much earlier and then RIG-I gets activated later during herpes viral infection. But a major question was, what are the RNA species that are sensed by RIG-I during herpes viral infection, or more generally during DNA virus infection? So again, presenting some of our earlier work, what we found when we purified RIG-I from herpes simplex virus or uninfected cells, and then did RNA-seq analysis on the portion of the RNA that was bound to RIG-I, we found initially, to our surprise, these were all human transcripts, but hardly any herpes viral transcripts being enriched or precipitated with RIG-I. When we zoomed into these cellular transcripts that bound to RIG-I during herpes simplex virus infection, they were all non-coding RNAs from different subclasses, here shown in different colors. And one of them stood out to us, here shown in red. They were highly enriched, and these were 5S ribosomal RNA pseudogene transcripts. So we all know, maybe I pause here, we all know, obviously the 5S ribosomal RNA, but what is maybe less well understood is that most species harbor quite a good number of so-called 5S ribosomal pseudogene transcripts. So they all have the gene name RNA5SP, P for pseudogene, and then different numbers. And prior to our work, there was really no role for them. It has long been believed they are DNA junk, and again, the role was not really well known. But I will show you in a few minutes. Our data indicate that they can be immunostimulatory in certain contexts, such as herpes viral infection, by activating RIG-I. What our early RNA-seq analysis also showed was that these pseudogene transcripts only bound to RIG-I when there was herpes virus infection, but not in the uninfected cell. And we could also confirm that for our top hit, RNA5SP141, and this was really the most consistently enriched RNA of these pseudogenes with RIG-I in the context of herpes simplex virus infection. We also confirmed that by qPCR, that it binds to RIG-I, but only when we infected cells with herpes virus, but not in an uninfected cell. What do we know about these pseudogenes of the 5S ribosomal RNA? I mentioned already functionally practically nothing, but we know that they are RNA polymerase III transcripts, and the RNA polymerase III leaves the triphosphate group at the 5' end. And furthermore, what we know through modeling, this is here a modeled structure of RNA5SP141. We know there are extensive double strand or stem loop structures, which led us to hypothesize that they might be indeed RIG-I bona fide agonists. Indeed, when we either in vitro transcribed RNA5SP141, we also did the same using a DNA construct and expressing it under a Pol3 promoter. So in either case, we found when you deliver them into cell or express them in cells, they're highly immunostimulatory, this RNA5SP141 pseudogene transcript. And when we silence RIG-I, the sensor, the response is ablated, but not when we silence MDA5. And we took along also a classical viral RNA, rabies virus leader RNA, and we consistently observed that at the same molar ratios, the pseudogene transcripts were even more highly immunostimulatory than the classical viral RNA at these concentration used. We can see the same if we assay for IRF3 dimerization by native page. So IRF3 will dimerize when there is interferon induction initiated, and we can see that these 5S ribosomal pseudogene transcripts efficiently induce IRF3 dimerization, similar to a viral bona fide agonist of RIG-I. When we remove the triphosphate group from the 5' end via in vitro phosphatase treatment, CYP treatment, this ablates really the immunostimulatory activity of these pseudogene transcripts. And the same is true for bona fide viral RIG-I agonist. But probably the most, or the gold standard in the field is using fully purified RIG-I protein, and then in vitro giving it either, the RIG-I agonist. So we gave it either the bona fide viral RIG-I agonist, or here this pseudogene transcript of the 5S ribosomal RNA. And then we measured ATP hydrolysis catalyzed by RIG-I. And you see that in both cases, viral or this novel host RIG-I agonist triggers efficiently ATP hydrolysis by RIG-I in vitro, which again indicated that this is a bona fide RIG-I agonist, these pseudogene transcripts. But we really wanted to know, how does this work? How does herpes simplex virus infection lead to the recognition of these pseudogene transcripts by RIG-I, and why is it not happening, in an uninfected cell? One clue to this question was what we found. There is a transcriptional induction. It is not massive. It's about 2 or 3-fold of this pseudogene transcript being transcriptionally induced upon herpes viral infection. We don't see any induction when we treat with interferon. So we think there's something happening really during herpes viral infection to induce the transcription. It's not a classical, ISG, what we can tell so far. But we also found, and I'm summarizing actually our published data here as a cartoon, we also found data that indicated a concept of unmasking. And I will tell you what we mean with that. So what we found is that in uninfected cells, these pseudogene transcripts are primarily contained in the nucleus. But during herpes viral infection, we found a massive relocalization from these pseudogene transcripts from the nucleus into the cytoplasm. And we think this is one key part of the mechanism of how they can stimulate RIG-I only during, viral infection, herpes viral infection. But what we also found is that herpes viruses, like many viruses, induce a host shutoff. So this can be transcriptional, also translational. Many times there are multiple mechanisms of host shutoff. But what we found is part of the host shutoff of herpes simplex virus, the virus downregulates certain RNA binding proteins shown here, TST and MRPL18, that we found normally mask these pseudogene transcripts in the cytoplasm. And we dubbed this RNA unmasking because upon downregulation of these binding partners, our data suggested that you get liberation of these pseudogene transcripts, allowing their binding and engagement with RIG-I to trigger interferon responses. Interestingly, you can kind of mimic the virus using an siRNA silencing approach. So we didn't infect cells with herpes virus, but instead we silenced TST and MRPL18. So these are the two RNA binding partners, and then delivered RNA5SP141 into the cytoplasm. And what you see when you silence and kind of mimic the host shutoff of the virus, you can strongly potentiate innate immunity, here shown by interferon beta transcripts, especially if you silence TST, this one binding partner, but also with the second one, MRPL18. We have generated by now knockout cells. These are CRISPR knockout cells where we completely excised RNA5SP141 from cells and then ask what is the innate immune response to herpes simplex virus infection. Consistent with our data using siRNAs and other measures to silence this pseudogene transcripts, we found in these knockout cells here shown two clones in red colors, there is a significant reduction or ablation of the interferon beta response. And we also measured, IFE2 as an ISG and CCL5 as well, especially at the later time points. So that means around 12, 16, 24 hours, here shown 16 and 24 hours post-infection. However, if you infect these cells with Sendai virus, that is a virus, it's an RNA virus sensed by RIG-I, and we know it's the viral RNA that is sensed in this case, we find there's no defect in the RIG-I or interferon response, which also ruled out that if you take out RNA5SP141 from the cells, it does not generally ablate, innate immunity. It seems very specific to herpes viruses. Mainly, I'm showing you today herpes simplex type 1 data, but we also have data on other herpes viruses such as EBV, Epstein-Barr virus. More recently, we found that in a family case of pediatric herpes simplex encephalitis, we found a mutation in the patient that led to a defect in the biogenesis, so the transcription of this RIG-I agonist, this pseudogene transcripts. So maybe a little bit of information about herpes simplex encephalitis, or HSE, how we call it. It's a very rare disease. It's a horrible and life-threatening disease where people can get encephalitis caused by either herpes simplex type 1 or herpes simplex type 2. And most prominently, it occurs in neonates and young children, but it can also happen in immunosuppressed individuals. It's very rare, but a devastating disease. In a portion of these cases, we know there are mutations in genes. We call them HSE susceptibility genes, and many of them are related actually to type 1 interferon responses. Most prominently, we know that certain mutations in TLR3, but again here listed other interferon-related genes, can be causative of herpes simplex encephalitis. The data, what I'm showing you, was a wonderful collaboration with Dr. Philomene Hernick's lab from Ghent University in Belgium, who actually experienced or had this patient of herpes simplex encephalitis. You see here the family, and the patient was a young boy that had developed early on a severe, herpes simplex type 1 induced encephalitis. And when Dr. Herring's lab did whole exon sequencing in the whole family, they surprisingly didn't find any mutations in the genes that I just showed you that were known susceptibility genes. But they found a new mutation in this factor called GTF3A. That's the gene name which encodes for transcription factor 3A. And there are two mutations. These are heterozygous compound mutations shown here. But a little bit about this gene or this protein that is encoded. So this is actually a cofactor of RNA polymerase 3. It's actually one of several cofactors of RNA polymerase 3. And it is involved in the transcription of ribosomal DNA genes, so making ribosomal RNA, and also the pseudogene transcripts. Again, there were two mutations that located to specific zinc fingers in this transcription factor, so in TF3A. And what we found is, without showing you the data for the first part, we found that there was no defect or anything in terms of mRNA expression in these patients who harbored the mutants. We couldn't see any difference in protein stability between the wild-type protein and this mutant protein encoded by the patients, and also not in nuclear translocation. But what we found, and this goes in hand with where the mutations sit in the zinc fingers, there is a deficiency of TF3A, how it binds to the DNA and to the promoter region. What do we know again about GTF3A, this cofactor of RNA polymerase 3? So first of all, we know that it mainly transcribes, for the most part, non-coding RNAs and only a few protein coding RNAs. And among the non-coding RNAs, it's really a large part are these ribosomal RNA genes or DNA genes, so encoding 5S ribosomal RNA, and then this larger part of pseudogenes, of which RNA5SP141, this RIG-I agonist, is one of several. Dr. Herring's lab performed an unbiased ChIP-seq analysis, and she found again that RNA5SP141, this RIG-I agonist I told you about, is one of the main targets of transcription factor 3A in this ChIP-seq assays. We looked at patient cells. We used fibroblasts and also PBMCs for our studies. We looked at the 5S ribosomal RNA, and there wasn't any difference between the patients. So this is the brother and the sister who has actually the same mutations as compared to healthy controls. And this wasn't maybe surprising. One would assume that, if there was any deficiency in 5S ribosomal RNA, that would, not be a viable phenotype. So this strongly suggests that there are compensatory mechanisms to still allow 5S ribosomal RNA to be made in these patients. But when we looked at RNA5SP141, this RIG-I agonist, we saw a significant reduction in the two patients harboring this mutation as compared to the healthy controls and also one of the parents. And it's not a complete ablation, but it's roughly a 60% reduction what we observed in these patients. We took primary dermal fibroblasts, again, from the two individuals harboring the mutations as well as healthy controls, and saw indeed that if you infect them with herpes simplex type 1, these individuals have impaired control of herpes virus, so the virus replicates significantly higher, here shown for 2 time points by plaque assay. And when we put back, so when we reconstituted these patient fibroblasts and put back wild-type transcription factor 3A, we could see repression of virus replication to about the similar level or comparable levels as the control cells. And indeed, we think that it has to do really this ablated control of the virus really with a type 1 interferon response. So when we analyzed interferon beta. We also measured multiple other cytokines and ISGs, which I didn't bring today, but here is shown interferon beta transcripts. These two individuals with the mutations in GTF3A have much lower, or an ablated innate immune response to the virus, to herpes simplex virus. Again, if you infect these cells with Sendai virus, a virus that is sensed by RIG-I, but where it's really the viral RNA being recognized by RIG-I, we don't see any ablated interferon response in the two individuals harboring the mutations. Just to mention it, I did not bring all the data, but we also tested poly-IC, LyoVec, LPS, and double-stranded DNA stimulation. So the patient fibroblast responded, comparably to the wild-type cells to all these different agents. And we also ruled out a TLR3 signaling defect in these cells as well. We also wanted to look directly at RIG-I activation. So RIG-I has multiple hallmarks of activation, one of which is this lysine 63 linked ubiquitination, which is a non-degradative ubiquitination type and has been shown by our labs and multiple other groups is important for its activation and helps the sensor to oligomerize. What we saw in these in the patient cells is when you infect with herpes simplex virus, RIG-I doesn't get lysine 63 linked ubiquitinated and also does not bind one of the E3 ligases TRIM25, here shown by endogenous RIG-IIP. But when you then exogenously give to these cells or transfect RNA5SP141, you can rescue the RIG-I activation seen here by efficient ubiquitination and TRIM25 binding. Just to mention, to rule out that there are any other defects in these patient cells, we also generated CRISPR knock-in cells where we put in these two mutations in GTF3A, here shown in the two green symbols. And indeed, these CRISPR knock-in cells harboring the patient mutations have a loss of control of herpes simplex virus, so the virus replicates much more efficiently, here shown by viral transcripts that we measured. And also we see this reduced production or transcription of RNA5SP141, this agonist, in the CRISPR knock-in cell, similar to the patient, what we saw in the patient fibroblasts. And when we interestingly compared herpes viral replication in these CRISPR knock-in cells compared to RIG-I knockout cells and also the knockout cells of this pseudogene transcript, we see very comparable virus replication. And that was interesting to us because it indicates that this one pseudogene, RNA5SP141, has a dominant role as a RIG-I agonist, at least in this context of herpes simplex virus infection, and contributing materially to that phenotype of controlling herpes simplex virus replication. So to summarize the first part of my talk, I showed you that RIG-I-mediated immunity can be triggered by signals that follow the pathogen assault, but originate from the host itself. And that's very similar to the GARD hypothesis in plant immunity. And our data really add to the larger emerging concept of host RNA sensing that has been shown now by multiple other groups in different contexts, that it's not only the pathogen RNA or the viral RNA, but in many cases, it can be unmasked, mislocalized, or misprocessed cellular RNAs that can trigger RIG-I-like receptors. And we showed it for viral infection, herpes viral infection. Other people have shown it for cancer or, of course, autoimmune diseases. And what our data indicate is that these pseudogene transcripts of the 5S ribosomal RNA indeed have a function, or in this case can be immunostimulatory in certain contexts. And I showed you as a last part that a defect in the GTF3A RNA5SP141 RIG-I axis can be associated with herpes simplex encephalitis. At least we have now one family where there was one case, and we will need to see if there may be more cases in the future that have mutations either in GTF3A or in RNA polymerase 3 or other parts of this axis. So now I would like to switch to the second part of my talk. I stay with the RIG-I-like receptors, but I want to talk a little bit about regulation of innate immune sensing, and we have done quite some work on that. This is kind of serves as an introduction. We know by now that just having an RNA PAMP present is not sufficient to activate innate immune sensing. In many cases, these sensors are regulated by different post-translational modifications, such as lysine 63 link ubiquitination, but there are many more, sumolation and phosphorylation, of course, and ADP ribosylation. And some of these PTMs, activate these sensors, and others will keep them inactive when they are not needed, either when there's no pathogen or at the end to kind of when the virus is cleared to kind of downregulate innate immunity. In the case of RIG-I, which is shown in this picture, and again, it's not really about the details, it's just to highlight how complex the activity of RIG-I is regulated by post-translational modifications. So what you see in essence here is in the center, again, the structure of RIG-I, the domain structure with a CART, helicase, and CTD. And you see how heavily this protein is decorated by different post-translational modifications. Phosphorylation, ubiquitination, different types, acetylation, and much more. And left and right are all the enzymes, so host enzymes, which either catalyze or remove these PTMs and thereby regulate the activity. I think it's fair to say that the most well-studied modification in RIG-I regulating it is this lysine 63 link ubiquitination in the CART, and multiple E3 ligases have been identified to catalyze this event. And going now back a while, we had the question how MDA5, especially in the CART, is regulated, and how this is really regulating its sensing and signaling activity. A while ago, we found using mass spectrometry that RIG-I, the CART domain, so these are the signaling modules, undergo ISGylation. So they're modified by ISG15, which is a ubiquitin-like protein. But what's unique about ISG15 is it's an interferon-inducible gene. So we always think about ISG15 being downstream of these innate sensors simply because it's induced by interferon. But what we found is it's really helping the sensors, so very upstream, to get activated. So we found this by mass spectrometry and then confirmed it by biochemistry, seeing always two major band and possibly also some additional sites in MDA5. We could show that if you stimulate MDA5 activity by either double-stranded RNA transfection, or when we infect it with flaviviruses, dengue or Zika, which are known to activate MDA5, we see an increase in the ISGylation levels of MDA5 in the infected cell. We also could see that when we use ISG15 knockout cells, these are mouse embryonic fibroblasts, and then launched RIG-I or MDA5 signalings, in this case simply by transfecting the proteins. We have similar data on using agonists and relying on the endogenous protein. In either case, we found that MDA5 signaling is completely ablated if you launch it in an ISG15 knockout cell, here shown by interferon beta ELISA. However, in the case of RIG-I, it can perfectly signal. There was no deficiency. And again, this was quite interesting to us. As I briefly mentioned, we always think about ISG15 being downstream simply because it is induced by interferon, but it actually indicates that it has a crucial role very upstream at the sensor level. And as our data indicated, it's by modifying the sensor MDA5 in the CART domain. We always detect by Western blot two prominent bands, which would indicate that there are two ISG sites in MDA5 CART. And through mutational analysis, we found that it's actually the two first lysins in the first CART, so lysine 23 and 43. When we mutated these ones, we could completely abrogate the ISGylation level in the CART. We also, I should say, mutated all the other ones by single site mutations where we saw no or very minimal effects. When we analyzed this MDA5 mutant, so the lysine 23R, lysine 43R, here shown by qPCR to measure cytokines or chemokines CCL5, we saw it was strongly ablated in signaling ability as compared to the wild-type MDA5. And without showing you here the data, the raw data, by now they have been published, we found that this ISGylation event in the card is actually important for MDA5 to oligomerize. And that is also, it's kind of similar in role or equivalent to the lysin 63-linked ubiquitination in RIG-I, where also this ubiquitination event helps the card to oligomerize in a similar role we propose for the ISGylation of MDA5 in the card. In data that is currently in revision, we generated MDA5, lysin 23R, lysin 43R knock-in mice. So these mice have only these two mutations. We used a CRISPR approach in that. We also, in the same approach and via non-homologous end joining, we generated in the same set not only knock-in mice, but also knock-out mice, which allow us now to really test the physiological role of ISGylation in an infected organism. And I should say for many PTMs of these RLRs, this hasn't been really done. So this was kind of exciting to us. What's really the role if we take out just the ISGylation of MDA5? What's the role in controlling virus infection and for the interferon response? In this case, we challenged the mice with EMCV, so that's encephalomyocarditis virus. It's a picorna virus that is primarily sensed by MDA5. And what we see is that in the MDA5 knockout mice, as well as our knock-in mice, which are MDA5 ISGylation deficient, these mice succumb to infection very rapidly. They cannot control the virus as compared to wild-type mice. When we measure the replication of the virus, so the titers either in the heart or then later the virus goes to the brain. Also in the brain, we see significantly higher replication in the knock-in as well as in the knock-out mice as compared to wild type. And when we measure interferon by various ways, here is shown an interferon beta ELISA, we don't see any interferon, at least not detectably, in the knock-in and knock-out mice. So this indicates that this ISGylation event is really critical in the card for initiating innate immunity by MDA5 and for controlling virus infection. This ISGylation event is also important for controlling other viruses. I showed you just the data from the knock-in mouse with picorna viral infection, but we also showed that it's also important to control Zika virus infection. This is here shown in SVGA astrocytes as well as SARS coronavirus 2 infection. So these are astrocytes knocked out of MDA5 where we put back a wild-type MDA5 or this ISGylation defective mutant. And you can see that wild-type can suppress Zika virus infection, but not the ISGylation defective MDA5. It's a similar titer of Zika as with a vector vector transfected or expressing, I should say, cells. And we also saw that whereas wild-type MDA5 could control SARS coronavirus 2 replication, the ISGylation defective MDA5 could no longer control it. But when we assayed all these different viruses, and we have even analyzed more than these here shown, we always get a quite robust repression of virus replication by the wild-type MDA5. With SARS coronavirus 2, we saw a quite marginal half of a log reduction, which led us to hypothesize that the virus might have found a way to kind of antagonize MDA5, and we ask if it specifically antagonizes the ISGylation event in MDA5. What we found is that the papain-like protease, PL-PRO, of SARS coronavirus 2 antagonizes MDA5 ISGylation. A little bit of background here. We know that PL-PRO, that has been shown by multiple groups for different coronaviruses, has both deubiquitinating and de-ISGylating activity. And these have been implicated in having immunomodulatory or suppressive functions. What is shown here is PL-PRO of SARS coronavirus 2, how it engages, here shown in blue with ISG15. And what we found, if we just express PL-PRO in cells, the viral enzyme removes effectively these upper bands or these ISGylation bands of MDA5 CARD. And this is really an active removal, because if we express C111A, that's a catalytically inactive mutant of the viral enzyme, this is no longer the case. And we also identified that this site 1 with these residues R166, E167 are important for this event as well, for this de-ISGylation. I mentioned briefly that we found that this MDA5 oligomerization is important. Sorry, the ISGylation is important for MDA5 to oligomerize. And consistent with this proposal, we found that when we expressed PL-Pro of SARS coronavirus 2, the wild type, this strongly suppressed the oligomer formation of the MDA5 CARD. And this is no longer the case if you mutate the catalytic activity in the viral PL-Pro enzyme. But what I haven't told you so far, so PL-Pro is only a tiny portion or a small portion of a really large protein of SARS coronavirus 2, that is the NSP3 protein, which is shown here. It's a really large molecular complex that is embedded in the DMV, the double membrane layer here, where the virus replicates inside these double membrane structure, or DMV structure, how we call it. Interestingly, the PL-Pro sits on the cytoplasmic phase. And what also has been suggested is that this large complex structure, this pore structure, is a putative RNA channel, where viral RNA can go through this channel. And this was kind of interesting to us, because MDA5 is an RNA sensor. One might hypothesize it wants to position itself very close to this pore where viral RNA will emerge. But our data indicate that PL-Pro, the virus, has already found a way to antagonize MDA5 using its PL-Pro enzymatic activity and de-ISGylating it. But what we found in SARS coronavirus authentically infected cells, we readily could detect an interaction between endogenous MDA5 and NSP3 of the virus. And when we looked at now the endogenous MDA5 ISGylation levels in the infected cell, we could hardly detect anything, indicating that there's a very efficient de-ISGylation happening by the virus on the endogenous MDA5 to keep interferon levels down. However, when we use the PL-Pro inhibitor GRL0617, we now could detect the ISGylation of endogenous MDA5, and this correlated with a restoration of the interferon and ISG response in these cells. But what I showed you, many of these data were derived with the PL-Pro protein only. My lab, we have expertise in generating recombinant viruses by ablating certain amino acids or certain host-virus interaction studies. So what we have most recently generated is a PL-Pro de-ISGylation deficient NSP3 mutant virus. So this PL-Pro is only deficient in the de-ISGylating activity, not in the deubiquitinating activity. And what we saw when we performed RNA-seq analysis, we compared this de-ISGylation NSP3 mutant virus versus the wild-type virus. This is in primary human bronchial epithelial cells. We see that the mutant virus is highly immunogenic, so it now highly induces ISGs. It's also attenuated, here indicated also by the RNA-seq, but you will see that in the next slide maybe better. So we generated this NSP3 mutant virus without a reporter. We also have one with a CS green reporter in each cases. We see here shown by qPCR that this mutant virus induces a heightened interferon and cytokine and ISG response as compared to the wild type, the parental virus. And this is also shown here in two different cells by Western blot. So if you take A519 human ACE2 cells or these primary bronchial epithelial cells, and then you infect them with a wild type SARS coronavirus 2, you see relatively little ISGs. Here by Western blots, we measure different ISGs. So the virus really is very, very good in keeping interferon and ISG levels down. But when we infected with our NSP3 mutant virus that is de-ISGylation defective, Now we see, the cells lighting up with ISGs, here shown by Western blot for the different time points, again indicating if you take out this immune evasion mechanism from the virus gets more immunogenic and is also attenuated in replication. And this brings me to the summary of my last part of the seminar today. So I showed you that the MDA5 card domain, the signaling module, is ISGylated. In unpublished work, we have identified it's mediated by the enzyme HERC5, one of the major ISGylating enzymes. And this ISGylation event in MDA5 helps MDA5 and the card to oligomerize and to allow the sensor to signal and to induce interferon. We believe that this MDA5 ISGylation event in the card has a functionally equivalent role to the lysin 63 linked ubiquitination in the RIG-I card. And what I also showed you is that at least one virus, SARS coronavirus 2, and we think it's broadly applicable to coronaviruses, antagonizes this step in the MDA5 activation pathway. I showed you that NSP3, which sits in the DMV, uses the PL-PRO enzymes to cleave off these ISGylations in the MDA5 card and thereby blocks interferon or suppresses interferon signaling. This is, of course, only one way of many ways how SARS coronavirus achieves this. And I showed you in newer work from my lab, we generated a PL-PRO de-ISGylation deficient NSP3 mutant virus that really then induced a much higher innate immune response and is attenuated as compared to the parental virus. Last but not least, I would like to acknowledge and highlight all the people who contributed to that work. I should say first, many people from my lab contributed to that. I'm highlighting here, the people leading these projects. So Jessica Chiang, a former graduate student from my lab, made the original discovery of these pseudogene transcripts being a RIG-I agonist during herpes viral infection. And then Leslie Naessens and Santoshi Mopala worked out, the mechanism and the molecular details in these patients, where the one boy developed a herpes encephalitis. And then Liu Liu and Chung-Yun Lee identified and worked on the project of ISGylation of MDA5 and defining the role in the MDA5 signaling pathway. And then Lucky Saka and Shunji Su are currently working on these projects. I'm lucky on this knock-in mouse, where the MDA5 ISGylation sites are ablated and really defining the role in an infected organism, and Shunji Su on this mutant virus, which cannot de-ISGylate to further define, the role of the de-ISGylation activity in SARS-2. I also would like to thank, our many collaborators, Philomene Herring from Ghent University in Belgium, Karl-Peter Hopfner from LMU in Munich, Klaus Osterrieder from the Freie University in Berlin, as well as Sean Stauffer and Bara Drosola and Susan Baker, who work with us on this SARS coronavirus 2 and MDA5 project. And with this, I would like to finish and again say, Katano, thank you so much again, for letting me speak today and inviting me. It was a pleasure, to be here and presenting our work. Thank you. Thanks.
Well, thank you very much, Michaela, for that beautiful talk. And again, the questions for Michaela, you can ask them via X or Twitter. The instructions are up on the screen right now. They were also on the chat box. And so it just remains for me to thank Michaela once again for her lecture. Thank you all very much for tuning in and for attending. Don't forget next week's Global IMMUNOTALK that will be given by Rino Rapwali. I hope to see as many of you there as possible, and also remind your friends that if they have missed this and any of the other Global IMMUNOTALKs, they are all available on YouTube, and you can watch them even if you've missed the live broadcast. And so with that, thank you all very much. Thank you again, Michaela, and see you all next time.
Thank you.